Hello viewers, welcome to our channel. Now we are learn about auxins.
Now we can study about all these hormones in detail. Number 1. Auxins. The word auxin was derived from a Greek word auxin, which means to grow, and they were the first plant hormones discovered. The term auxin was introduced by Kogel and Hagen-Smith in the year 1931. Discovery of major plant hormones began with Darwin's experiment on phototropism. In the year 1880, both Charles Darwin and his son Francis Darwin conducted three experiments on phototropism. They observed the effects of unidirectional light on canary grass coleoptiles. Look at the canary grass. Do you know what are coleoptiles? Coleoptiles are the protective covering or sheets present on the tip of the grass. During their experiment, they noted that the coleoptile was very sensitive and responsive to light. Look at this image. In the first experiment, they observed young shoots bent towards the unidirectional light source. In the second experiment, the coleoptile was covered with an opaque foil cap and noticed that bending did not occur. And in the third experiment, an opaque collar was placed below the tip, and this time bending occurred but was not as usual. And they came to the conclusion that some influence was transmitted from the tip of the coleoptile to the basal region, but they couldn't name it. Later, Wendt, a Dutch biologist, identified this influence as auxin. Now we can study about Wendt's experiment. Fritz Warmolt Wendt was a Dutch biologist who demonstrated the existence and the effects of the growth hormone auxins in plants. Wendt experimented with young shoots of odd plants. Avena sativum is the scientific name of odd plant. At first, he cut off the coleoptile from a shoot of odd plant and noticed that the plant stopped its growth. In the second experiment, he placed an agar block, or we can say a gel block, on the decapitated coleoptile tips. Decapitated coleoptile tip means the tip of the coleoptile that was removed. This time, the coleoptile did not show any response. So, in his next experiment, he placed the detached coleoptile tips on agar blocks. Detached coleoptile or cut tip of coleoptile that you can see in this image. After 1 hour, he placed the agar blocks on the decapitated coleoptile, and this time he noticed that the plant grew straight up. So he concluded that some chemical had diffused from the cut portion of the coleoptile's tip into the agar blocks, and that stimulated the growth. And finally he named it as auxin, which means to grow. Types of auxins. Auxins are of two types, natural auxins and synthetic auxins. Natural auxins are produced by the plants. Example IAA, that is indole 3-acetic acid, whereas synthetic auxins are the auxins that are artificially prepared. Example 2,4-D, 2,4-dichchlorophenoxyacetic acid.